Follow us on social media and please hit the subscribe button so you don't miss out on any updates from our ALS experts.
It is so nice that every 2 weeks we find our extended family, especially in one place. And you all are just, from 8 inches across, I'm getting to see a whole array of beautiful people. So I just want to say welcome to our meeting this evening. And whatever you feel inspired by that you hear tonight, talk to us later in the open mic and tell us what you learned. So with that, Lisa, welcome.
Thank you, McFinn. Welcome, everyone. Thank you so much for being here today for our Everything ALS Wednesday webinar series. We are so excited to bring the experts directly to your living room. So I'm Lisa Deegan, and I am part of the Everything ALS team. My younger brother had ALS, and each one of our team members have personally been affected by this disease. So we're all here to help you out, and our goal is to provide you with what we didn't have when we were going through our journeys. So tonight we are looking forward to an evening in education on the latest research from our expert, Dr. Clive Spensen from Cedars-Sinai. He's going to be presenting on a new California Institute for Regenerative Medicine funded clinical trial, transplanting stem cells, releasing GDNF into the motor cortex of patients with ALS. After the presentation, as McFinn mentioned, we're going to do Q&A, but then we're going to follow up with a non-recorded private session to let members meet and connect with everyone in our community and talk about whatever's on your mind. So we'll look forward to that as well. Before we get started, I quickly want to bring up our I hope that everybody can sign up for our voice project to help end ALS. With the help of everybody, we can bring together citizen-driven research along with data technology and science to help with getting an earlier diagnosis of ALS and a better understanding of the course of this disease. So the goal is to get treatments accelerated. So please visit everythingals.org/research to learn more about this. And please consider signing up. Only takes about 5 to 10 minutes a week, and you just need to be 18 and over. And I think Sarah had talked about this earlier. So hopefully you got to hear some of the criteria. I also want to mention briefly that Everything ALS has a new podcast called Stories and Innovation in ALS. And you can catch this on Spotify and on YouTube. And we share in our website. So we're sharing stories of those affected and also those who are working so hard to find a cure for this. So if you are interested in sharing your story or your work, please email me, lisa@everythingals.org. We'd love to share and highlight your story. So now I'd like to introduce tonight's presenter, Dr. Clive Spensen. He is the Executive Director of Board of Governors Regenerative Medicine Institute, a Carrie Simone Vicker Family Foundation Distinguished Chair in Regenerative Medicine. He's Director of Board of Governors Regenerative Medicine Institute, and also a Professor of Medicine and Biomedical Sciences. Dr. Spensen has a long-standing interest in ALS, and he was the sponsor of the first ever clinical trial delivering stem cells and GDNF to patients that was recently completed at Cedars-Sinai. So he founded the Cedars-Sinai Board of Governors Regenerative Medicine Institute, whose main focus is to model and treat various human diseases with the use of induced pluripotent stem cells, also known as iPSCs. So Dr. Spensen maintains a large lab that focuses on modeling neurodegenerative diseases, including ALS, SMA, Huntington's, Parkinson's, and Alzheimer's. Using orphan-on-chip technology that combines stem cells and engineering, he's creating a multicellular human system for research and drug development. He's got many recognitions for his work, including a recent feature on the front of the cover of National Geographic as the future of medicine. So please join me now in welcoming Dr. Spensen. We appreciate all the work you do for ALS and your dedication and showing up tonight to share it with our community.
Cool. All right. Well, thanks, everybody. Very nice to be here. It's been a very busy day, as Hyundai knows. The Niels meeting was this morning, where there was 400 or 500 people on the call. As you know, the NEALS is the East Coast Consortium that does a lot of the clinical trials in ALS, and we heard from Merit and many people that you know about. So big day, but this is even bigger. Very excited to be part of everything ALS again. In fact, this is my second time, so this is kind of my talk two. I did want to direct you to this, which is the original talk I gave. And at this point, I talk more about stem cell trials in general. And right now, I'm going to discuss a very specific trial. But you can go back and rewatch that if you can. It really fills you in on a lot of the basics of stem cell biology and as it relates to ALS. There's been some other great talks. One here from Nathan, who went into mesenchymal cells and their treatment potentially for ALS. And then Namita gave a great talk on the neuron trial, which I'll mention as I go through as well, which is just completed, as you know, the phase 3. And then finally, we can't forget Stan Appel, the grandfather of this field. And he's got some amazing work going on using stem cells peripherally administered to treat ALS as well. So I have to say it's a very exciting point in the field and a very exciting time because there are some new options coming up using stem cells to treat diseases. And I encourage you to go look at these. The wonderful thing about everything ALS is you can go back and relax, have a cup of coffee, and go watch Hyundai again and just explaining about the upper motor cortex and Stan explaining about his treatments, because they're all a little bit different. There's been, since my last talk, just one slide on some progress. We were waiting desperately for the outcome of the Brainstorm trial. This is where they put mesenchymal cells, and these are cells they take from your bone marrow, expand, and put back into your brain, the CSF surrounding the brain. This trial has read out, and unfortunately, it didn't meet its endpoint had to be a certain amount successful to meet its statistical endpoint, and so we're all very disappointed about that. But they did show that the cells were taking out some bad things and putting out some good things. So there's some hope there that this trial has taught us something about putting these cells into the spinal, into the CSF. And again, I'll come back to this trial a little later on. And there's some very exciting work going on in Japan, where in my previous talk, I talked about iPS cells, and you've probably heard about this technology where you can generate stem cells from a patient's own skin and then reprogram them, take the skin cells, reprogram them to be stem cells. And because they're from ALS patients, you can use those to test drugs. And in fact, there's a study in Japan that's just come out showing that you can use these to generate ideas for what drugs might work in specific groups of patients. And there's some hope there. It's very early days that by using this technology, we can use stem cells in the dish to predict drug effects. So those are the two exciting sort of events that are happening. They'll be talked about a bit more at the NEALS meeting, I think. Aside from that, those two reports, not much else is very new in the stem cell field. But there has been progress in the trial that I talked about last time, and I'm going to update you on that first. And then I'm going to tell you about a new trial where we've moved from the original target where we were putting the cells to the brain, and then I'll conclude. So this is the idea. Many of you have seen something like this. It's a horrible slide with lots and lots of information. And it really tells you that ALS is a complex disease, right? And it involves lots of different pathways. But essentially, this is the motor neuron in the cortex, and this is the motor neuron in the spinal cord. And Charcot, who discovered the disease, said right at the beginning, this is two diseases. It's a disease of the upper motor neuron, and it's a disease of the second motor neuron in your spinal cord that projects to the muscle. Both those neurons are depicted here. This is the upper and this is the lower motor neuron. What I'm going to focus on in this talk is how we might be able to help, because there's an idea that, yes, the motor neuron's important, but the motor neuron has nursing cells called astrocytes, this guy here, that sits next to these neurons and protects them and actually stops them from degenerating in certain circumstances, and that these cells may well be going wrong in ALS, these support cells or astrocytes. And it's the astrocyte that we're focusing on. So imagine you could replace these with new astrocytes, what would happen? And that's the question we asked. And then the second part of this is this drug called GDNF, which is kind of an amazing compound. It's a growth factor. That means, if you know amyotrophic lateral sclerosis, the trophic part means lack of trophic factor is part of the disease. Well, this is a trophic factor that we can now deliver to those dying motor neurons using a technology that I'll describe in a moment. And this trophic factor has been shown to protect motor neurons from cell death. So this is a combined product that we've developed. And the hypothesis that we're testing is these transplantation of these astrocytes will slow down motor neuron death. And here's the two-part disease, the lower and the upper. And I'm going to discuss first this part, which is the lower motor neuron down here. And how can we replace the astrocytes next to those lower motor neurons that project the neuron out, the axon to the muscle? How can we maybe inject cells here and replace the astrocytes in ALS? And that was really the first trial that we did. The last cartoon, just to put this home, here's the intact system with the astrocyte, the motor neuron, and the muscle. We know in ALS something goes wrong, and we think could be to do with the astrocytes in addition to the motor neuron, but becomes abnormal. And then the motor neuron pulls away from the muscle in the spinal cord. So the idea is to replace those astrocytes. And a lot of work that we've done has shown that astrocytes on their own are good, but it's even better if they can make GDNF, and we engineer the cells to make GDNF. So now this is the combined astrocyte and GDNF therapy. So where do we get the cells from? Well, we can isolate these cells from brain tissue, very, very early age human brain tissue. We can expand them as these balls of cells, we call them neurospheres. And then we add a virus. And normally you don't like viruses, particularly COVID, but believe me, viruses can be very useful. And in this case, we used a virus, a lentivirus, that encoded GDNF. So now when you infect the cells, these neural progenitors from the brain, you expand them, and they're now engineered, if you like, to make GDNF, this protein that's good for ALS. And then these cells can go on to make astrocytes. So we now have a product, if you like, where we can manufacture these cells. And one fetal sample will give enough for 4,000 or 5,000 patients. We can get a lot of expansion out from this one sample using expansion technology that we have. And in fact, we've a lot of work to do this in a way that's available to patients. This did take about 4 years of work, understanding how to grow these cells, engineer them to make the GDNF, but that's all done. The product is called CNS10 NPC GDNF, because it's a CNS10. It's actually my initials, Clive Neil Spensen, but it's also central nervous system. And it was the 10th line that we looked at, and it's neural progenitors, NPC, and GDNF is the growth factor. And this was a while ago. Here I'm going to bring in the state of California. There's quite a few people here on the call from California. They did a wonderful thing. They gave $3 billion back in 2007 for the state of California to do stem cell research. And we got one of the first large grants that allowed us $18 million to do all the work that you need to do to get to the clinic. That included animal studies, making the cell lot, so making the cells, as I just mentioned, animal studies to show it's safe, and then finally the clinical trial. And we've had a whole team of people I mentioned at the end who have been very helpful with this. Well, how does it really work? Here are cells making GDNF. And what we actually do in the animal models is take a pipette and with a very sharp needle on the end and basically inject those cells right into the spinal cord of these SOD1 rats. And this is a model of ALS. I think Hande might have mentioned in her talk, we need animal models, and in these animals, the motor neurons die. You can see here, this is a normal spinal cord, and this is a spinal cord where there are no motor neurons left. And this is the area we're going to be focusing on today. And these spinal cord motor neurons die over time, but if we put the stem cells in, releasing GDNF, and the animals become paralyzed, the first thing they do is they will integrate into the spinal cord. The second thing they do is make GDNF. The brown staining here, the brown marker, is GDNF protein in the spinal cord. I think you can see this is the whole spinal cord, this is the right side, and this is the left side. But we put the cells in just on one side, and it's basically producing GDNF for the whole side of that cord. And when we look at the cells themselves, this is in red, these are the individual human cells in a rodent brain. They've actually migrated around the dying motor neurons. And what we find is they can protect the motor neurons. So up is good here in this graph, and these motor neurons are being protected in this animal model of ALS. So this gave us all the proof that we needed that these cells could go in and protect motor neurons in ALS. And that moved us to a lot of different papers along the way. This is going all the way back to the first papers, but I'm not going to go through any of these today. Just to show you that we actually showed they could protect neurons. We actually have done this in the areas of breathing as well, in the cervical cord, although we haven't done a trial there yet in humans. They've survived long-term. It didn't form any tumors. And they also actually worked in older animals. It's a question we always get asked. So we actually did a study where we put these cells into old rats, and they still seem to differentiate and protect motor neurons, even in old rats, which really helped because a lot of patients are a little older. We also put these cells into pigs in order to look at a larger animal. And you can see this black staining here showing we have 10 sites in the pig. And this was what the FDA really liked. We could, with 10 injections, because the cells migrate, cover the whole of this side of the spinal cord in 10 sites at 2 millimeters apart. So that covers 2 centimeters of the spinal cord, and 2 centimeters is enough to cover the whole lumbar region that controls your leg movement. So that really was the road to the IND. This is the small document that we submitted. It was 4,500 or plus pages. We got the IND approved and moved to the actual clinical trial. This trial was completed very recently, and I'm going to update you on it, and then I'm going to move to the second part, which is for the cortical. Now, remember, this is one site in the lumbar spinal cord, so in the part of the spinal cord that provides motor neuron support to your legs only, and we only did single side. Did 2 different doses, 9 patients at the first dose and 9 patients at the second dose. We used immune suppression. These patients, as we have to. This is a product that could reject, although we have it from this trial, we've now understood a lot more, and I'll tell you about that in a moment. And it was unique in that we only gave the cells to one side of the spinal cord, so that the other side of the spinal cord almost served as control, so we can understand, do these cells actually do anything in terms of slowing progression in the legs that we treated. Primary outcome measure, that means what was the task, what was the trial going to show? For instance, in the previous trial I mentioned from Brainstorm, their primary outcome measure was a certain amount of efficacy. Our primary outcome measure here was actually just serious adverse events, so safety. And I can tell you now, 18 patients, a very complex trial, it did meet its safety primary outcome, and we're just about to report that back to the FDA. The secondary measures were just to see what happened with this first shot at strength testing and other aspects, and also to collect the tissues. We did lose patients within this trial. They came to post-mortem, and we've looked to see, the big question for everybody is, did the cell survive? And did they make GDNF in a human spinal cord? So that was the second question. And here's how we do the injections. It's a laminectomy. Patients under complete anesthesia. The neurosurgeon goes in with a small needle, looks for the location, injects the cells, and essentially the cells then will do the rest of the work. They migrate out from where we put them, and they go to the area of those motor neurons that you can see there in the spinal cord of the patient. The neurosurgeon ties everything back, and the patient is usually discharged within 3 days. The only thing really keeping them in the hospital is the immunosuppression. And then once that's completed, the first round of infusions, the patient can go home. This is truly a one and done. Once the cells get into the spinal cord, they integrate and become part of the spinal cord. And so they are really cells which will last the lifetime of the patient should they survive, and in that way should be able to protect the motor neurons for a long period of time as well. That's the concept. We did the first patient back in 17. It went very successfully, as did the next 18. This is Pat Johnson, I'm pointing out in the center there, and Pablo Avalos and my team. And it was a group of a large number of people that made this happen. And the surgeries were very efficient, all went successfully, and every patient was discharged without any complications from the surgery. We were very happy when the last patient was dosed in October. And then we had the readout a year later from the last patient, November 19, and we all know what happened. Right after that, COVID hit, and we've been desperately struggling to try and get all of the bits and pieces together from this trial and collect the data in this rather complex environment, but we are doing very well in terms of this. And as I mentioned, the primary outcome is achieved, and we're now closing these documents out this month. The report is ready to send. In fact, it's sitting on my desk downstairs. It's my next job. Once the FDA get that report, the trial is officially closed, and we can move to the next phase. And what happened? I mean, obviously, we got safety, so that was good. What happened to the patients? We've shown previously that this device is called, some of you may have been in one, it's an Atlas. It's an isometric system. It measures your strength. And we've really found this is a very accurate way to measure decline in function over time. As a patient gets weaker, the strength gets weaker. And that's a good clinical outcome. And it's turned out to be very reliable. So that if you see an effect in one leg, you might see that that leg becomes stronger over time or stops becoming weak over time. And so we use this based on a paper that we published to show that most patients, both legs go at the same speed. One might start before the other, but then they tend to go at the same speed of loss of muscle strength. And so we wanted to know, did the side we transplanted go slower? Now, I'm not going to go through all the data because I presented this before, and I really want to focus on the new trial. But I will tell you, on average, we saw in the high dose group, 0 here means there was no effect. It means both legs progressed at the same speed. Anything above the line means that the leg we treated did better. And the low dose is this green dot here. So there was really no effect of the low dose, good or bad. And we remember, this could have made it worse as well as better. This is the first time we've tried this. So there was no bad effect. And in fact, at 12 months in the low dose, we saw a significant improvement. This star means it reached significance. But there's only one time point at the 12 months, and that was the end of the trial, that we were monitoring the patients. We continued to follow them after that, but that was the end of the official trial. At the high dose, we saw a good increase over time, but nothing reached significance. So we took away from this that we are having a positive, minimal positive effect. We definitely aren't doing anything bad. And the details of the trial are a lot of the patients, by the time we enrolled them, because it's safety, were already quite progressed in their disease. And so we think, as one of the conclusions of this, we need to go a little earlier in the disease. And we did have one other problem, and I'll tell you that in a moment. And that was the targeting, as I showed you earlier on. We were trying to target an area where the motor neurons were, obviously, to protect them. So the second outcome was, where do we hit? Now, I'm going to show you one slide here. And this is GDNF in patient 104 who came to postmortem. And in this case, we really had the perfect targeting. Here's a group of cells, and you can see how they've spread out the GDNF and the cells right throughout the motor area, and are now there to protect the motor neurons in this region. So we're very happy with this case. It seemed like even around there were a few more cells surviving, but unfortunately that was only in one site or two sites in this patient. Many of the other sites were located a little higher, and in many of the other patients, the sites were located higher. So all of the patients had transplants, but the location in most patients was a little bit too high. The cells didn't migrate down quite as far as we thought. And so the next thing is basically, I'll tell you in a moment, to go a little bit deeper in our transplants, but we've definitely learned that where we put them there is safe, and that when they do get into the right place, it's safe as well, because those patients did very well in terms of safety, and we may be having some small effect on motor neuron survival, even though the patient was late in the disease. So safe patient, the treatment was safe. We had that trend in improvement. We saw some neuromas, which is a slight swelling and growth around where we injected the cells. I think this is because of a little bit of trauma to the input to the spinal cord, but they were benign and didn't cause any issues. And as I mentioned, the transplants were a little bit too high in most of the patients, but a few we hit exactly right. So where are we going to go? We're now cleared. Once COVID restrictions are lifted, we're going to go a little earlier in disease progression. We think that's going to help get a better effect, but certainly target a little deeper to hit the ventral hole directly. We know we can hit it now. We know we can hit it safely. Now we're going to be a bit more aggressive in the next trial. This is how phase clinical trials normally go. You start with a very modest attempt, very careful, go high so you don't interrupt the neurons that you're trying to actually protect. Then you go more aggressive if you show you can hit them and it's working. And now we're going to be going a little deeper in the spinal cord. Now that's all fine, and the spinal cord is obviously very important in ALS. But really the last part of my talk, I'm going to shift gears a little bit and explain to you, based on that success, that the cells survive, they make GDNF, and we're going to go back and target a little differently. Even if we got it exactly right, even if it was fantastic, would we still slow progression? And the argument is, yes, maybe you'd help, but don't forget, we're putting the cells down here in the lumbar spinal cord. And all the way up here, are the motor neurons which actually enable you to initiate movement. So what if those motor neurons died, even though you protected the lower ones? That's always been worrying to me as we've gone through, moving these studies into the clinic. And so I've been very interested in this upper motor neuron for a long time. And in parallel to all of the clinical trials, I've been working hard, moving the research forward, try to understand more about this. And the reason we're interested is, as many of you know, maybe you've had this technique, transcranial magnetic stimulation. There is this effect in ALS of hypersensitivity early on. So if you stimulate in ALS, you actually get a faster response early on. It's like your cortex is firing too much early on in the disease. And this comes before the late stage. And the late stage is actually this decreases, but early there's this hyperexcitability in the motor cortex, which is very common in ALS. And we think this may be driving some of the cell death of the motor neurons that I'm trying to protect. I told you about, we're trying to protect down here. There's this lovely review here about this process and how ALS patients may have a cortical involvement. There's somewhat of a, I wouldn't call it an argument, I call it a discussion in the field about where ALS starts. And in my life, it's not it's there or here, it's what is the evidence for this versus this? Because we don't know the exact answer to this. But there is a hypothesis that the upper motor neurons in the cortex are very important and they may actually drive the rest of the disease. Now, we don't know if that's true or not, but there's evidence to support that. And I think it's interesting in that riluzole, which is the only drug that works, actually blocks the release of glutamate, and that these motor cortex neurons actually release glutamate into the spinal cord motor neurons to excite them to fire to contract the muscle. So what if riluzole was actually blocking an excitatory input to those motor neurons that were actually being driven to death by the upper motor cortex that had gone wrong? And then you play the game of, well, what's causing the upper motor neuron problem? Well, it could be actually neurons projecting into those neurons. That's a whole other story I don't have time to go into today. But it's a hypothesis I think definitely needs to be tested. And of course, we have Hyundai on today. I grabbed a screenshot from her talk that I listened to 3 times last night, so I'd make sure I wouldn't make any mistakes today, talking about the upper motor neurons. It's a wonderful description of her work, which really, she is a pioneer in this area for understanding the pathophysiology of the upper motor neurons. They become atrophic and they lose some of these spines, and the circuit dysfunction is very important in early ALS. And so Hyundai, in our questions, can maybe talk more to that, but it's the brain is early in disease as well, as she points out here. In some beautiful imaging studies further supporting upper motor neurons. This one from Aziz, and Aziz has shown in these cases, the inflammation is shown in this PET scan. So all around this motor cortex here, you can see this yellow area showing that the motor cortex is not right, it's inflamed, because it's picking up this marker that you don't see in control patients. And the controls are down here at the bottom. You can see they're nice and clean, whereas in ALS, you get this inflammation. And you see this more in the upper, when there's an upper component, but you also see it when there's a limb component, although not so much with bulbar onset patients. So this upper motor neuron is involved in ALS. We're pretty sure of that. And I think the imaging actually is producing a fantastic new way to study this, and imaging studies are really taking off, and I think predict they're going to be very important for ALS coming up. Just 1 or 2 papers from our lab. This is really the work of Gretchen Thompson, who's an amazing postdoc in my lab and has now gone on. I think she's working with Brian Caspar now. She went to Avexis for a while. And she's basically shown that these cells, that you can take a mouse, that in this case, a rat model of ALS, that I mentioned before with this mutation, and you can knock down, take the mutation out just in the cortex, the motor cortex, which is this green area here. We labeled it with GFP. And if you take out the toxic protein, the SOD1 that you overexpress, you make too much of this protein, it kills the neurons and gives you ALS. If you take it out just from the upper motor neurons, and this little lack of red here means the protein's disappeared, but leave it everywhere else, all through the rest of the cortex, but also into the lower motor neurons, magically, you stop the rodents from progressing. So the red is the animals that had the treatment, the knockdown of the SOD1, and the blue is the animals that didn't. So you can see they continue to become paralyzed on this score of paralysis. So the shRNAi, which is a way of knocking down SOD1, has continually protected them. And so that was just in the motor cortex. So that told us the motor cortex is important. At the same time, GDNF is known to protect upper motor neurons as well in models of axotomy and a number of other different models. So that combined product, astrocytes and GDNF, should work in the motor cortex. So then we did the obvious experiment, again, Gretchen Thompson. This is with ALS Finding a Cure, Leo Zutto Foundation, that I'm a member of the board, and so is Merit and a number of others, and they really helped progress this. And then we got funding from the DoD to continue these studies. And this kind of funding is really important, seed funding for these kind of experiments, because this led, as I'll tell you in a moment, to a very large grant, thanks to ALS Finding a Cure and the donors that came forward to support this work early. So Gretchen got together with my team and asked a question: if we put these GDNF, CNS10 GDNF that I told you about, that we already had ready for the clinical trial, into the motor cortex only, what would happen in the ALS rat? So here are the cells making GDNF. You can see GDNF staining. You can target beautifully this part of the motor cortex. Again, we didn't put them anywhere else. And I won't go through all the data, just to show on these slides in red is the treated animals. And you can see they've come out in time, so they're not dying as fast, and their progression is much slower. And this is just cells put into the motor cortex. And we didn't, really fine tune the dosing in this study, but we showed there's a nice, strong effect, behavioral effect. And in the paper, we actually show that it's a very strong effect on motor neuron survival as well in the spinal cord. I don't have time to tell you. So that means by slowing down the degeneration of the upper motor neuron, you've actually helped the lower motor neuron as well. So this was kind of very interesting to us. And actually this data, to be frank, was stronger than the data we had when we just put the cells in the lower spinal cord, where the function was more difficult to see. We got protection of motor neurons, but the functional changes were not as strong as we saw when we put them into the upper motor neurons. Further supporting the idea, Hyundai's idea, and others, that the upper motor neurons are actually very important for driving disease progression. Now, we have a lot more work to show and prove that, but it's definitely being supported by this data. So then we jumped forward. We said to the FDA, look, we'd like to go into the upper motor neuron cortex. And we then had to get another grant from the DoD for a lot of animal studies, about $5 million worth of animal studies and 2 years of work. We just completed all of those studies. We showed we can get beautiful GDNF administration in GLP study, which means it's FDA kind of compliant study in a large number of rats. And you can see how beautifully the cells integrate. You see the transplant but basically the cells migrate and they release GDNF. And these brown cells here are motor neurons, large motor neurons that have taken up the GDNF and being protected from the GDNF, but at the same time have the astrocytes that migrate around them. So we did that. It was a clean study. By the way, with those neuromas I mentioned we saw in the spinal cord, we never saw neuromas, these small balls at the entry site of the spinal cord in the cortex, because the cortex doesn't have this dorsal root entry zone, the area that caused the problem in the spinal cord. So we don't see these artifacts, these neuromas in the motor cortex. So this is actually a little simpler for our study. We then did primate studies as FDA required. We put the cells into the motor cortex of monkey. And this was with Chris Bankovich, and the study was clean. We didn't see any bad effects in the monkeys. We followed them very carefully, and that was further data supporting the move to the clinic. And based on all of that, we then looked to how do we translate this to the motor cortex. So another 5 or 10 minutes and I'll be done. And I'll finish up with this description of what we're going to do in the humans. And we're very excited to present this, and somewhat for the first time to the patient community. So this is what the human motor cortex looks like. It's a fascinating structure. As some of you might know, it's this weird thing called a homunculus. Now, this is your brain, the right side of the left side of your brain. This strip here is the motor cortex that drives all of your limbs. This is what goes wrong in ALS, this strip. Motor neurons die, become dysfunctional. So where are we going to put the cells? It's a long strip, right? I mean, neurosurgeons could actually do lots of sites and cover this whole area. They're very gung-ho. They can do a lot of surgery, and there's actually, neurosurgery is now becoming common in diseases like Parkinson's. So it's quite standard now to have DBS, which is they put an electrode into your brain and it works quite nicely. And hundreds and more thousands of Parkinson's patients have this neurosurgery. So this isn't something that is rare. It's a lot of neurosurgery goes on in neurodegenerative diseases. It's not very scary. It's a small hole to drill, and then they insert the cells. So we had to choose, though, which part of the motor cortex. We chose to go with the hand area. So the motor cortex is led, if you go from top to bottom, you start with your toes and your feet, And then as you go around this purple area, you move through all of your body lenses. It's somatotopically mapped onto the cortex, called a homunculus. In fact, a large area down here is devoted to the face and jaws and eye movements because of how important that is. I'm speaking right now because of this mapping of the lower part, but also a very large part obviously goes to the hands. The ability to do fine movement is all driven in this hand motor knob. So we've analyzed this carefully with a neurosurgical team led by Adam Mamalak at Cedars-Sinai. We are putting together we've put together the protocol. The rather nice thing with the motor cortex and these surgeons is they know how to find areas of the brain through stimulation. They can put small electrodes, they can measure activity. And in fact, we can actually measure the activity as we put a probe in and decide how deep to go and find that layer 5 where the motor neurons are based on the electrical recordings. We've mapped out in postmortem tissue how we're going to approach this. We're doing 21 sites, and this is the surface of the cortex, and these are little dots that we've been using to mimic the injections that we'll do in the patients. And again, we've mapped this out. We've done primate studies, and we're ready to go for a patient trial. So a lot of this is based on the work we've done in the initial IND, and in fact, this is covered under our initial IND. So it just required more data to be submitted to the FDA. We submitted that data. They did not issue a hold notice, which means we're approved to go ahead. And the IRB was approved. COVID spike, again, we've been waiting, but now we're clear in LA. The numbers are low enough so that we can go ahead and initiate the trial. Now, we were waiting for funding. We applied to CIRM for our 3rd round. And we were very happy to hear just in June this year, end of June, that we received enough funding, 12 million, to do this full trial that I'm going to describe now. This trial is, again, phase 1 safety. Now we're in the motor cortex. Nobody's ever put cells in the motor cortex before. We will be the first to do this in patients. We want to assess safety, first of all. So we'll start small. We have a total population of 16, but we're going to start with a small number of patients, just 3, and we're going to do that in the non-dominant hemisphere. Now, the cortex, of course, you may not know this, but you have 2 brains. We have a right and a left. The right part of the motor cortex projects through to the left part of the body. So when we put the cells on this side, we'll expect an effect on the opposite side, the opposite hand in this case, because we're going for the hand knob area. We'll start small with N of 3. What we'll do is the first patient, we then stop for a month, watch very carefully to see if there's any negative effects before we're allowed to then move to the next patient. And this is a very closely monitored trial. If it's safe for the first patient, we go to the second and the third, and then we app the number of cells. And initially, we're going to use the non-dominant hand. So if there is any bad effect that we didn't predict, you've affected the hand that's not used for writing. And then finally, or anything else. So then finally, we'll start the larger part of the trial, which is group C, where we go ahead and do 10 patients with the dominant hand in the 21 sites at the higher cell dose, because we figure that cell dose for our animal studies is probably the perfect one. These patients will be suppressed. I have to tell you, from the first trial, we suppressed everybody for 1 year. And then some patients passed away 3 years after that, or 2 years after the last immunosuppression. And everyone except 1 patient had a surviving transplant, even though they'd been off suppression for almost 2 years in some cases. So I think the suppression works very nicely, the immune suppression. We may even be able to get it down to 6 months less, because we have some evidence that might work as well. But for now, we're going to immunosuppress, get the cells engrafted. Once they're in, you can remove the suppression after a year, and the cells will continue to survive. We're also going to be testing with this 9-hole peg, because we're really focusing now on dexterity and hand movement. I'll talk about that in a moment. What's fascinating about this part of the brain is there's a lot of very new papers and exciting papers coming out, which I think are relevant for ALS generally, about this hand knob area that controls hand function. So we're leaning into this. We're talking to the authors of these papers, understanding how we can really focus in on this area of the motor cortex. And Hande will be excited by this, but just by recording in patients before we get to put the cells in, we may start understanding about regions which are becoming less active or more active as we progress through with this trial. So it has a lot of side features, this trial, that we're going to learn a lot about, I think, the upper motor cortex in ALS patients. It may even be that the area that we target, we think is the hand, we're now discovering it's more complicated and covers other areas. So this paper suggests that some of these areas of the hand knob actually have a wider implication across the body. So by saving motor neurons here if we can, we may have a wider effect. Now that's obviously rose-tinted glasses, and we don't know for sure, but that's something else we're excited to see from the outcome of this trial, what happens. And finally, the test you'll love, very simple, 9-hole peg test. How long does it take you to do the simple test? It's not electronic, it's not handheld, it's the old-fashioned 9-hole pin test. It's turned out quite a good predictor of progression and hand dexterity and function. And so Rich Lewis, who's the PI, clinical PI, will be administering this to the patients in this trial. So not too bad, not too laborious, and kind of fun. So we hope to do the first patient November 2021. We're pushing hard. There are a lot of kind of things to get done before we get there, but we're moving along nicely, and I'm predicting we should be able to get clear and done by November 2021. So to conclude, and I'll then stop for questions, I just want to refer to the Brainstorm trial and how this is different. That's a question I often get. Brainstorm, as I mentioned, uses adult cells derived from the bone marrow of the same patient. We use fetal-derived cells, one line that is then administered to all the patients. So it's not what this is called autologous, the Brainstorm technology, because it's from the same patient. Ours is just one source of cells that goes into all patients. The Brainstorm cells are injected into the CSF, the cerebral spinal fluid, via a lumbar puncture, and that then gets in around the fluid, the CSF it's called, cerebral spinal fluid that surrounds the brain. And then the mechanism is they probably take out good things, bad things from the CSF and maybe release growth factors, one of which we know is GDNF, which might help, but they really, after a while, don't survive. So you have to re-administer and redose, which is fine. If it becomes effective and if they move forward and they find an efficacy with a different dose, then they can keep dosing. And unfortunately, as I told you, the first trial didn't quite meet its endpoints, and they're looking for ways to continue those studies. And we can talk more about that in the discussion. The key point is that those cells we know don't integrate into the spinal cord or brain, and they don't turn into a tissue of any sort. Whereas the description of the trial I just showed you, we know our cells, the ones that made from fetal tissue, will make new astrocytes in the spinal cord, and we know also in the motor cortex. So this is essentially a one and done. I don't like that term necessarily, but it makes the point that, okay, the surgery is pretty invasive, but once you've done it, actually you don't need to take a drug even. Once the immunosuppression is done, you're done with taking the immunosuppression. The cells are in, they release the GDNF, and you don't have to take anything, no pills, nothing. So in theory, these cells would last the lifetime of a patient. They're fetal, they'll outlast the patient if they integrate and survive, which we think we've shown from our first trial in the spinal cord. So we're excited but cautious. I'm always, this disease is one of the toughest. It's very hard. And so not saying we've reached a cure of any sort, but we've definitely reached a big milestone moving up to the cortex. I think based on Hyundai's work and some of the data I showed you, that this may be a very exciting target to try this out in. And I have to say, for me, this has been a marathon and not a sprint. There isn't a miracle cure just 2 seconds away. It's often a lot of work. Like the first heart transplants failed terribly. Eventually, those transplants now become common. And I think our first transplants we're gonna be asking questions, learning from each trial, but we have to keep moving forward. And the advantage of doing it carefully and us getting safety means we now are allowed to do the next one. And it's usually science or research, whenever you do something, it raises more questions like our first trial did. But the advantage of being safe and cautious is now you can do the next thing and the next thing. And I think that's where we're building on our initial results, and we're really hoping that these cells will have some sort of functional effect in the disease. And as we all know, anything is going to be great, and we're really hoping that we can get protection and slow down progression in patients. There's been many people involved with this. I couldn't thank everybody enough. Just in this picture, we have Pat Johnson, who's the neurosurgeon, and Chirag Patel. Many people from my lab, pharmacy, all over the Sinai. And somehow here, very importantly, is my wife, Shana, who was the only one who read the 4,500 pages of the IND. She's a fantastic colleague in addition to my wife. And somehow my daughter slipped in here, Nicole, who's blurry because she's probably racing from one side of the room to the other. This is a few years ago now. But this is the actual team. And again, just thanks to CIRM in particular for the funding and just the refunding again. And to all our other funders that I mentioned, ALS Finding a Cure, the ALS Association and the initial, some of the very early work we did with this, with Lucy Bruin, was kicked off by the ALS Association. We're very excited, again, to move this forward. But most of all, I'd like to thank the patients who enrolled, because we told them, we're very clear that you're just injecting cells into one side of your leg and this isn't a cure, we're not. And yet they went and they enrolled in the study because they knew that we'd learn from this and move forward so that patients in the future may be able to benefit from a larger trial or going into more sites. So the key is you guys and your participation. And I will stop there and I will take some questions.
Absolutely. Well, first, Dr. Spensen, thank you so much for joining us again. I am Sarah. We spoke the last time, but that was a while ago. So I am an Everything ALS team member. I'm a med student at University of Michigan, and I am just over the moon excited about this talk. I'm talking to my James, who's also going to be doing questions with me, and we're just like, we're geeking out right now. So thank you so much for presenting, because this has been amazing. But we will get started so we don't waste your time. And you kind of mentioned it at the end, but how could current people going through ALS potentially get involved in this research in the future?
So you can contact the clinic. I should probably put a number in the chat. This new trial is just being set up, so we don't have a hotline quite yet, but I can give it to Everything ALS and let you know. For now, I could drop my email in the chat, and if people are interested in enrolling and participating, I will put you in touch with our ALS clinic, and that's Dr. Lewis. So the other way is to look at the Cedars-Sinai ALS clinic. It's an ALSA-funded center of excellence. Rich Lewis is the neurologist there who's going to be the PI on this trial.
Wonderful. Is there any restrictions to where people live in order to participate?
Yeah, we like people to be close to Los Angeles or in California at least, because of the testing and the movement in and out of the clinic. So our preference is South Carolina, Southern California. See, I'm British, I don't know my job. Southern California. Having said that, yeah, and I know that's painful for people who are on this call who live elsewhere, but that's, as you all know, it's the nature of these trials, because you have to come back every month or two for analysis. It's a lot of work if you live outside of California, and it's a single site trial right now. If we're successful, we'll obviously open it up to more, and there are some patients who, will do that traveling and can afford it and will do it, and so we will consider those, but our preference is local.
Thank you.
We always have a lot of people willing to jump in for these trials. So I'm glad to hear that we have some contact information for that. To continue on that line of thought, though, are you allowing patients in the late stages of the disease into the next phase of the trial?
So the inclusion criteria are quite specific for this trial, and it's we do this because we want to see some hope for some effect. And if too late, then we're not going to see anything from our secondary outcome measures. So just like all other ALS trials. Now, I think it's the other way around. I think especially Hyundai can chip in here, but I think with the upper motor neurons in particular, it seems like they go through a phase of dysfunction before they die. With the lower motor neurons, it seems like they crash. If that's true, if we can show safety and maybe a little functional effect going into mid-stage patients, we might want to go more end stage or further down, because you might restore function. If the neuron is still there and connected, you may be able to kick it in with some more GDNF or an astrocyte. Same as the drug studies that Hyundai is looking at. I have more hope that the upper motor neuron may respond later, but that's the second part. I think that's a follow-on trial if you show safety and a little efficacy in this first. I wouldn't say the word efficacy, but safety is important. But some hint of something happening would be nice as well. That's why we're doing it.
Absolutely. And Hyundai, I have allowed you to be able to unmute yourself. So if you ever want to jump in, we'd be happy to hear from you too. Our next question, for those that maybe not know, can you explain a little bit more about what an astrocyte actually is and what you mean by it protects the motor neurons, or it protects the neurons?
Yeah, so an astrocyte is like being at the doctor. If you're at the doctor's office, you have a doctor and who really does nothing, and then you have the nurse that really treats you, right? The astrocyte is the nurse. It's a nursing cell. Motor neurons die, right, and they crumble. But all around the motor neurons are astrocytes. There's 3 or 4 of these astrocytes. They're cells. They've got lots of processes, and they kind of tickle the motor neuron, and they stay beside it, and they nurse it to make sure it's healthy. And there's some evidence that those die. Now imagine losing the nurses in a hospital, the patients would all die, right? So it's the same thing in the brain. If you lose the astrocytes, those beautiful motor neuron chandeliers, will unfortunately not function. So they're the support cells. They don't get talked about a lot. People are obsessed with neurons. I love astrocytes. They're important. They look after your brain cells, keep them healthy, the neurons.
Well, shout out to all the RNs out there.
I'm gonna be careful. I'm in a hospital, so I love the doctors as well. I love everybody.
Well, I'm a nurse too, so I started out in nursing, so it was definitely. I'll shout out to both, but thank you.
So the next question was one of mine. I was trying to understand as we were going along. So if I heard correctly, is the hypothesis that the astrocytes themselves provide some type of nourishment for the central nervous system? And if that is the case, what nutrients are suspected to be lacking for those with ALS?
Yeah, so the astrocytes do a lot, right? So we call this a combined therapy because you could just put the astrocytes in and get very excited about that. People do get excited because they don't only produce nice factors, but they actually absorb things that are bad, toxins from around the neurons as well. They also form part of the blood-brain barrier. And the blood-brain barrier, we know, may be also deficient in ALS. It's very controversial, to be honest, but it could be. So by replacing that, you improve the blood-brain barrier, you make the motor neuron happier, and it's kind of an overall health. Now, the reason we do GDNF in addition is when we did our initial studies in rat models, we didn't get a good effect with astrocytes on their own. It wasn't as impressive, but when we combined it with releasing this drug right around where the astrocytes were, boom, we had a nice positive effect. So I'm like, that's a no-brainer. I'm going to make them produce GDNF. Why would I put, right? We have another trial going on for retinitis pigmentosa. We did our first patient 2 weeks ago. We haven't announced it yet. We're going to wait until patient 3. There the cells are just going in the back of the eye and they make an astrocyte layer. Doesn't release GDNF. We didn't engineer them. Different function for the cells. These cells are quite plastic. They make astrocytes in the CNS. They make a different type of cell in the eye, in the back of the eye. But we've got this product moving now in different indications, which is exciting.
Wow, I'm just so excited about this. Is there any current evidence that shows certain environmental factors may damage or kill these astrocytes? And if so, which ones?
Alcohol, no gin and tonics. No, the environment, see, the brain is so buffered by the environment. It's a great question. In fact, I got asked that this morning. Slightly different question, but similar. In ALS, you get this toxic protein you've all heard of called TDP-43. It's like amyloid for Alzheimer's. It seems to build up. So one of the questions I had this morning in the Niels talk was, what if TDP-43 leaks in from the patient brain and damages the astrocytes? It's a possibility, right? My feeling is they're young, they're healthy, they're 0 years old. I know I felt really good at 0 years old. I'm just turned 60 now, so figure they have a lot of energy, so they're going to cope with that much better than the astrocytes that are already there. These are rejuvenated astrocytes, they're brand new, and so they should be able to cope. The environment, the environment of the CNS is kind of its own environment, and so I don't think environmental factors should affect it. I'm a big believer in 1 gin and tonic a day.
There we go.
A little bit of everything.
I kind of party.
Yeah, I can handle one. So on the same line of thought, are there any other concerns for other types of glial cells to be damaged? And, just again, could you maybe explain what the overall branching glial cells are?
So there's 2 types of glial cells there's 3 types of glial cells in the brain. The one is the astrocyte, which I was talking about. The second one is an oligodendrocyte. But that's really important too. That's in all the white matter tracts that run up and down. So the oligodendrocyte wraps around the motor neuron as it goes through the lateral columns. And it's called amyotrophic lateral sclerosis because that represents those oligodendrocytes that ensheath those cells going down die, and you get that lateral horrible changes in the lateral columns. That's the second type of glial cell, an oligodendrocyte. It's for myelination. That's the one that goes wrong in multiple sclerosis. MS is a degeneration of those types of glial cells. The third one is a microglia, and that's a cell that's in the brain. It's like an immune cell. And the microglia get very angry if things are bad, and they react and they mop up things and clear debris up. So those are the three types of cell. Now, you can replace any one of those. Jeff Rothstein has a whole program on oligodendrocytes that he's shown that those are defective as well, a little bit in ALS. There's microglial inflammation. So we've seen lots of different glia, but I'm just focusing on the astrocyte because that's the one we know we can restore, and it has probably one of the major functions in maintaining neuronal health.
That's really interesting. Kind of switching gears a little bit, but in regards to the immunosuppression, you discussed how long patients would have to be on it. Is there any long-term concerns or even short-term concerns during that time?
Patients actually cope pretty well with the immunosuppression. I mean, a few had to come off. They just couldn't tolerate. I think 2 of our patients came off at about 4 months and 6 months. The other 18 maintained it to the year. And boy, they were happy the year that they didn't have to take it anymore. But it's something that, they actually tried it in ALS trials because they thought, well, maybe if we suppress the immune system, we'll stop inflammation, which Stan Appel talked about, and help the disease. And it turns out just doing that doesn't make any difference or impact on ALS. There was a clinical trial that tried it. So on its own, it doesn't affect the disease. People take immune suppression if you have a heart transplant or a liver transplant, kidney, you have to take it the rest of your life. So a year is not too bad.
In, again, regards to the immunosuppression, is there any way to incorporate cells from the patients themselves to avoid the immunosuppression altogether? Specifically, you stated that the fetal cells turn into all types of cells, but is there any way to make patients' own cells with the same ability?
Yeah, so we have, and I didn't have time to talk about it. I can talk for 5 hours, by the way, if you want me to keep going on a talk with it. Okay, so we've got this is the iPhone 1, all right? The iPhone 5 I'm on right now. iPhone 1 is what we're putting into patients because it's reliable, and it's somewhere in the iPhone 3, we've got a way of taking your cells, right? So we could take your cells, James. Anybody's on this call, your skin cells, we can put them in a petri dish. And as I said in my last talk, and Jeff Rothstein's talked about this and others, we can take them back in time to make them pluripotent. Now they're your own cells, right? So, and we found that we can make neuroprogenitors from those cells as well, those iPS cells, which are your own. And we've got a whole program, and we're just about to publish a paper that shows that those cells actually work pretty well. Now, but they're not as good as the fetal-derived astrocytes. We've tested them one-on-one. So the old solid iPhone 1 is still working. It's like getting an iPhone 5 that has glitches in it. And you're like, why did I get this thing? You throw it away. I'm not moving to that until we're happy that the fetal-derived cells use those, because those are the ones I know work. They're safe. The iPS-derived ones are technically very cool. You wouldn't need immunosuppression, presumably, because they're from your own body. But we still got to just work out the glitches in the software, if you like, to get them perfect.
Your analogies make this so easy, so thank you for this.
Sometimes they go astray, so please stop if they go off track and I get down a whole rabbit hole and I get in trouble. So
you're all right. All right. The next question kind of talks about the dosing. So you said that the higher dose in the spinal cord showed a little bit more of a directionality and was considered safe. Are you planning to use that dose in the upper motor neurons in the brain, or are you planning to use a different dose?
It's a similar dose, yeah, similar dose per injection site. And I think we were actually very happy in the spinal cord with the dosing. Like I said, what we didn't expect was our targeting was a little higher than we wanted. We were being cautious. We didn't actually want to go too low and actually disrupt those delicate motor neurons. And so we started a little high, hoping that the cells would migrate down. And from the postmortem data, they didn't migrate down as far as we expected. And so that's why the next patients were going to go deeper. But a few did, and a few looked beautiful, and they've hit right on the target. But the same dose is the answer.
In the hyperexcitability portion of the disease, has there been any research to determine that early of tracking it? And if so, could it be used as a diagnosis?
Yeah, so that transmagnetic stimulation I talked about was developed. It's been used primarily at a group in Australia, but there are other groups using it. It's been used in a few clinical trials to follow progression. The challenge is it's a little unreliable. It's great. I'll use another analogy. It's a bit like a Ferrari. It's great when it runs, but when it goes wrong, it's horrible. And I think some of that technology is trying to get consistent results from patient to patient has been the challenge. But if you average out, it's great. There's definitely hyperexcitability. But if you incorporate into a clinical trial, it's been a little challenging to get patients into this machine and get them to stay still long enough to, actually do the analysis and get it to be reproducible over time. So it's one of these finicky assays that has a lot of potential, but it's been difficult to industrialize it across a lot of clinical trials. We've actually had a lot of debate, Richard Lewis and I and the whole team, as to whether we should include it in our trial. And we're just a little too worried that it's a lot of encumbersome on both the clinicians and the patients, and that we're not sure we're going to get enough back from it, given that there is hyperexcitability, but it varies quite a lot from patient to patient. And so we're probably not going to use it as a biomarker for this study.
I don't know if you see this hyperexcitability prior to disease onset, or because cortical excitation, the Australian group see it about 6 months prior to disease onset, and then it goes to hypoexcitability, right? So the cortex response to treatment may be detected with the TMS method. It's a potential. It's a potential,
but as you know, Han, it's a little tricky.
It is.
To even get it before onset, because how do you know it's before onset if you haven't got it yet? So it's a tricky one, but it definitely changes from hypo to hypo very quickly. So as the motor neuron gets sick after a few months or a year, now it's not, it's dying, it's like, and so it ends up not functioning at all. It's like a car spluttering, the engine's overfiring, and then it's gone. God, I'm really going for the analogies today. Okay.
Our next question kind of talks about going back to the astrocytes again. Is there any way to measure these in people, whether they have ALS or not? And is there any difference in their astrocytes between people with sporadic or familial ALS?
That's a great question. We need more post-mortem studies, Sunday, right? We need more ways of looking, because you can't really see these until you get a post-mortem sample from a patient. And one of the challenges we have in the field with post-mortem data is that's the end of the disease, right? So you don't know what happened to the astrocyte when you first got the disease, and you just can't get that data. I mean, so how do you understand what's happening early on? Then we go to animal models, and then we get to that whole thing of like, there's no animal model of sporadic ALS. There's only an animal model of SOD1 or C9. The C9 model doesn't even show anything really that like patients do. So there's a lot of problems with models. So it's been a challenge to answer that question honestly. We don't know really what happens to these astrocytes early in ALS, and that's the answer. We don't really know. We're assuming they're going bad, and there's animal studies that suggest if you damage the astrocytes, the motor neurons then die, or if you express SOD1 just in astrocytes, motor neurons get sick, which is giving us, that's why we think they're important in the disease.
It's just incredible. So for the future, if the human studies do go well in the hand, would it maybe be possible to put those said cells based on a different location, example for those who have bulbar versus limb onset?
So yes is the answer. I have some very excited neurosurgeons that I'm always holding back. They really could go up and down that. Remember that strip I showed you? And literally you could hit, the foot is right on the inside of the surface. We actually initially thought, let's go for the foot because we've already got a leg and foot function. But it's a little harder to target for the neurosurgeons. You've got to go down into the midsection of the brain and go in. They said the hand knob's exposed, it's an easier section to transplant. So a lot of this stuff is practicality, but yes, if we get safety, if we get any efficacy in the hand region, we can then start going up and down. Bulbar's a little complicated, but we could also think about some of those areas do have an effect on bulbar, but bulbar's a little more complicated. I don't want to go into that now, but it's a little bit more complicated.
Okay. In terms of the study where the SOD1 protein was knocked out, how exactly does that happen? And would it ever be possible to maybe consider that in humans?
Well, funny you should say that. Bob Brown has a very nice paper. He just talked about it this morning, actually, at the Neil's meeting. So the answer is yes. So if you happen to have the SOD1 mutation, you're one of the 3% of ALS patients that has that mutation. There's a trial coming near you, maybe in the future. And he used the technology is it's called shRNAi. Horrible, right? What does that mean? Well, basically, it stops the protein being made. SOD1 it's an overexpression. So you make too much of this nasty protein in your motor neurons and they die. So you use this virus and it puts in this gene which shuts down that nasty protein production. That's the shRNAi. What it means is that it stops the RNA from making protein. So, oh, the protein goes down and wow, you clear out the SOD1 accumulation. Bob Brown did one patient. It was published in the New England Journal of Medicine recently. The patient there was some complications like every first trial, some pain, other issues, but it looked promising. And they're now doing another 3 patients, I believe. Hyundai, you were at that talk from Bob, but they're moving forward to try and get distribution a bit better. And for ALS, if you have SOD1, you should be contacting studies like that are trying these really unique trials. Because when you know the target, I said this the last time, if you know the target that's causing your disease, you should be enrolled in trials that are focusing on that target, whether it's C9 or SOD1 or TBK, whatever your mutation is, which is why I'm encouraging everybody to get genetic screening. We had some great work this morning about that. Why not know in case you're in that category? There's only 12% of patients have genetic mutations we know about. The other 88% are what we call sporadic, right? But we know that of those sporadic cases, about 40 to 50% is inherited, which means it's passed on some way, diluted down. And that was the talk this morning, was all about how do we understand more about that large chunk that the genetics play a role, but it's very weak. And it's probably now the environment that adds on the stressor. You asked about stressors to the astrocytes. Most ALS is probably caused by a genetic weakness followed by an environmental toxin of some sort that we don't understand. And that's the key. How do we work that out? That's what a lot of research is going on right now to understand. Sorry, I'm waffling on. I should shut up.
No, this is, I think, fantastic. It's so encouraging to hear how much progress and time and dedication is coming out of this. This is excellent. But we do want to be respectful of your time. So I believe this may be our last question. We hear about stem cells a lot, but just to kind of break it down, could you perhaps explain how exactly they are physically injected into a patient's body? What is that process?
Sure. We actually have it's not too difficult. We manufacture the cells, we put them into a little tube about that big. The neurosurgeon has imagine an injection needle that you got your COVID vaccine from. You suck it up into an injection needle, and we literally push it into the spinal cord tissue and inject it. And the cells squirt out in a blob within the spinal cord. Then we pull the injection needle out and then seal it all up. And the cells now become alive in that spinal cord and integrate and migrate a little bit, and they form the transplant. That's how we do it. And we do the same in the motor cortex. So you're putting a minute bunch of cells into the brain. It's kind of like science fiction, right? The cells you put into the brain, though, you have to do the invasiveness, right? You can't do a lumbar puncture like brainstorm. This is invasive because we've got to get the cells into the brain tissue where the motor neurons are dying. We don't want to put them all around because they don't get in. Now that involves a needle. It's scary. We don't like needles. You're asleep. Neurosurgeons know what they're doing. They put them and they target those areas that Hyundai mentioned, the areas that are dying with the neural progenitor. And we call them progenitor because they're not mature yet. They're a baby stem cell or a baby astrocyte, let's say. Once they see that environment, they migrate little distances and then they stop, and then they put their branches out around the motor neurons that are dying. And then they're in there, they're integrated, they're chimeric, if you like. And that's what we're trying to achieve. I hope that explains it a bit better.
Absolutely. Well, thank you so much for your time this evening. It has been an amazing presentation with so much information. I have written down so many notes on things that I need to go read and understand and rewatch, but I am so grateful. And I know everybody here, we obviously would love to have you back as many times as you want to come present.
I think 2 is my limit. I don't want to hold the stage, so I will stop at 2, but I really love this program. I think you guys, I love this community. We're all working, all of us, Hyundai and I, and all the researchers working for you guys. That's the only reason I do this. So love to all of you, and we're trying hard.
Yes. Well, thank you so much.